Hey guys, okay, so as part of our this collection of videos, I'm gonna run through just a quick overview of how to process single cell data in DIANN. And of course, you're probably familiar with DIANN, and I have nothing to do with DIANN at all, except for as a user. And of course, this is when we first heard of this. This is from Vadim and out of Marcus Rausser's lab, and there's actually a great follow-up study where they use single cells in specific methods. But this is the reference, and this is how we process data in DIANN and how we visualize it. So the basic steps here are first to acquire bulk data, and bulks and quotation marks here because that can mean many different things. Either we, under specific conditions where we have multiple conditions, we may make a really nice for example, S-trapped, homogenized, lysate, we fully quantified. It has all of our conditions, and we will run small amounts of that on the same system where we run the SCP. And in other cases where this doesn't make quite as much sense, where we're collecting single cells, it's common for us to have say 100 cells or 200 cells, depending on the instrument, single FACS sorted cells put into the same well of a dish where we perform, where those are digested under the same conditions as single cells, and then we run those with a DIA method, and then we process that through DIANN. We do this so we can create a sample-specific spectral library. So we're not doing a library free search for the entire, human proteome. This is really helpful, and there's multiple papers here showing that, yeah, library free is an amazing thing, but nothing. We're still at this point where nothing really beats having a sample specific library generated on your instrument under the same conditions. We find very, very large gains here. I think this example set that I'm going to show you, we don't really see that here with this DIANN analysis, but in the other videos, we'll see where this is. Then of course we just make sure that sample is good. We don't want to waste time on generating a library, and we really want to dig into that and make sure that it's representative and of the sample we want to actually analyze. Do we see the proteins we expect? Maybe we cross-reference with the protein atlas. We have that benefit of being using human all the time, so we can go to those expression profiles and make sure that we are seeing that we aren't measuring a large library of contaminants, right? Then, and normally we do this in the same batch off of the same plate, especially if we're FACS sorting, for example, 100 cells into, into A1, blanks into some wells, and then single cells in the rest of them. But normally we acquire the single cell data under the closest conditions possible. We have found detrimental effects when we acquire our single cell data from a bulk cell lysate on one instrument, and then we apply our single cell data on another instrument, even from the same manufacturer, some specific alterations in these hardware configurations. You want to get those conditions as close as possible. And then we process that single cell data against exclusively that new library. And there's the great reasons for this. So the first one is that if you're doing a single cell study, which I'll show in some of the other videos where it's like a label free of 200 cells, it can take forever to do library free search. You get this big, not only do you get a more specific and increased data completeness by using this library, but it's way, way faster to process this against the library instead of predicting a completely new one every single time, right? And then what I'm going to show you is just how to take the protein groups matrix and load that into SCP Viz so we can start to make sense of the single cell data we've generated, right? So that's the end of the slide deck. Let me just exit out of this, and I'll show you just some other things in DIANN. Chances are you've probably used DIANN more than I have, but just the things to go over here if you're not really familiar with this and are using it in this context. First of all, we just want to pull up a diapassive study. I'm going to go with this, and again, I know that in this experiment that in every case, file B is 100 FACS sorted single cells, and I could tell whether that worked or not in general by the size of the file here. B1 is, nearly 4 gigs. And if you look at B2, B3, which are actually single cells, these are like a gig and a half. This is pretty common when we're looking this at DIANN. But here, I'll start with this. But realistically, I've processed, control, and treated in multiple replicates to make my actual library. You want to clear out your spectral library here and just start from scratch with one without a name, because I'm going to generate a library. And then, since at this point I don't have a library, I am going to do a library free search, and I'm just going to use the Uniprot reviewed FASTA, the more, most recently from 2022. Um, at this point, you do want to use FASTA Digest, to get your library generation, where you're going to do the deep learning, and then set your normal conditions here. Chances are, if you're doing single cell, I guess it depends. But the majority of the new methods really aren't doing reduction and alkylation, because it's really hard to get that those reducing agents and things back out. They impact the instrument downstream and the tryptic digestion and things, because we just can't get them out of the single cells. Most people aren't doing that. So then you turn off the alkylation. Um, um, haven't really tried in DIANN to look for phosphos, but here, just set these to normal things. I haven't noticed any differences in actually changing my M over Z range. It seems like DIANN's smart enough to realize that that's what it is looking at. What's really important here is you actually want to make sure that you are generating a spectral library. This isn't always selected by default, and you want to tell it where to put that library. If you do not change this, it will copy over the last library you generated. We've had cycles of users who are getting used to the software, and they repeatedly copy over their own libraries. And then basically, here's my bulk data. I'm going to run this, and I'm going to generate this spectral library. Right. So then when you go back to the spectral library, clear these out. Now I've actually got a library to choose from. And here in this case, we'll just pretend that I hadn't selected this out. This is my HStack library, non-reduced control cells library that was generated for another study. But here it is. It's here. At this point, I do not want to do prediction. I do not want to do any of this deep learning stuff. I just want to read the library that I've already generated that has this information in it. And point, yes, you can generate a spectral library from your single cell data, but if you don't change this output, it will again copy over your results from your original spectral library. So if you do want to keep that, most of the time I don't really care what the spectral library is from the single cells, because the bulk it's going to be a small fraction of what we see in the bulk. And then I just select my output where I want it to go. Then when I add in my single cells, I could just add these in here and have it search specifically against my bulk spectral library. And as you see here, again, these runs didn't seem to work very well. This wasn't a fantastic set of runs, but in general, look at this. We're finishing these runs on DIANN. I think the entire length of this searched when I did not have to do a library prediction. It was just against the spectral library. We're talking like 20 minutes or something, right? Here we went cross-run analysis thing. So yeah, maybe this was 15, 20 minutes to run from nearly 1 o'clock to 1:30. And if you do this, if you take these same 12 cells and you run them and you generate the library, you may be looking at 6 to 8 hours on a 16-core computer like this. Obviously much slower, much faster on a better computer than this. I should note, right? I am using ultra fast because I'm really realistically using DIANN for QC on this actual computer in the lab. But you probably do want to run this for optimal results, so that you're actually getting a deeper profile on each single cell. Now, we go here. What you'll get in your output directory here is like temp directory, main output is here in this report. I think you can find that. It's in the DIANN demo. Okay. So normally we would be looking at, we'll often just go ahead and open the protein reports, but what we actually want to see is a protein groups matrix, the reports, PG matrix. First pass is, yeah, it's a little bit smaller because it's not the second pass through on DIANN. But if we look at this, what we get is this output, and this is important here because we'll need to know what some of these columns are so we can actually look at these data. But what's important here is if you are going to use SCP Viz to look at your single cell data or to evaluate it, you can't use a TSV file. You need to say this is a CSV, but it's really simple. Just go ahead and save this as, demo .csv there, and you can find it somewhere here, just a regular CFV, and you can save it, right? Okay, so I'm going to assume if you don't want to use SCP Viz to look at all this data, go ahead and use whatever you want. If you do want to use SCP Viz, we just want to get SCP Viz loaded up. And actually, I was looking at this data, I forgot to cancel it out. But here, go ahead and load my demo data we just generated, that CSV. And what SCP Viz needs is just to know where the protein abundance or peptide abundance, if you're doing peptide specific visualization, but it just needs to know what columns is it looking for. And since these are my quant columns across, and every one of them has the word DIA, has DIA in them, and none of my other columns do, I just said, okay, well, I'm just going to kind of parse this and tell SCP Viz that DIA is actually where my protein abundances are. Then it needs to know what you want to use for your protein identifier, the protein that you actually want to look at. And here, we're going to use protein group. Now, what's important here is that R is pretty bad at dealing with decimal points unless you have specific packages installed. So what SCP Viz does is recognizes any of the decimal points or periods in your CSV, converts them to an underscore. So what you want to look here, and if you have any questions, you can just go in this pull down and say, okay, well, my protein group's ID is here. If you're more comfortable working with genes, you can go ahead and choose the genes column, and then you can start pulling up your genes that you know. Here, I just went ahead and looked at a protein that had, was detected in multiple single cells, and I just copy and paste it in here, right? So I just copy paste. Wait, I'm not going to do that on a Mac. Yeah, but I could just copy that in here. Again, I could just take any one of these protein groups across, and then I tell it how many conditions. Since this is just a demo, I go back and I just choose a modifier that is in all of these columns. And since 30 minutes is in every one of my quant columns and it isn't in any of the other columns, all I have to do is put that in there and unleash the analysis. And so my summary table will give me a breakdown of for this protein, whatever it is, what's the max, mean, median, et cetera. I can bar plot it. What I processed here in these 12 files, B1 is actually, or what is this? Or no, C1. C1 is actually 100 single cells. So it makes sense that this protein is being seen in 100, significantly higher than any of the single cells across the board. We could go ahead and generate a PCA plot. That does take a minute, depending on the computer, and especially if you're running SCP Viz on our Shiny app server, which it only dedicates one core to this process. It takes a very long time, but we can go across and start to develop these. There'll be more on how SCP Viz works, but I just wanted to show you how to get data processed in DIANN and get that into SCP Viz. So that's all the goals for this video. Thanks for listening.